Thank you for joining us tonight. My name is Piero Scaruffi. This program is sponsored by the Stanford Deans of Engineering, Humanities and Sciences, and Medicine, by Chemical Engineering, and by Continuing Studies. LASER stands for Leonardo Art Science Evening Rendezvous. It's been going on since 2008. Over the years, we've had more than 400 speakers, usually in person on campus. And there are laser series now in more than 50 universities worldwide. Go to leonardo.info if you want to know about lasers in other locations. Go to lasertalks.com if you want to know more about the series that I personally chair. And at lasertalks.com, you can find the full bios of our speakers and links to their website. We have 4 independent presentations today. And so I will introduce each speaker separately, actually. You can see on the screen where the speakers, and you probably already know if you are logging in. So without further ado, we can start with the first speaker, Mike Jewett, who joins us from Stanford. Apparently you're outside, not far from all of us, but Mike, so my introduction is very brief. Go to lasertalks.com if you want the full bio and the link to the speaker's website, which is a lot more information. Michael Jewett is professor of bioengineering at Stanford University. He conducts research in synthetic biology for manufacturing, medicine first, manufacturing, sustainability, and education, and he's the co-founder of several startups. Okay, Michael, if you want, perfect.
Wonderful. Just a quick sound check here. Are we okay?
I think if you speak a little louder, it's better for the video recording, but yes.
For sure. So
okay,
thanks again.
I'm going to kill my video and my audio to give you maximum bandwidth. So I ask Kat and Will to do the same, please.
Okay. Thanks again for the fantastic opportunity to be here to present to you and everyone about our research in synthetic biology for people and planet health. Before I start, I need to make mention that I have financial interest in some companies that could potentially benefit from our research. But what I'd really like to talk with everyone today about is the use of biology as a general purpose technology. So our research group works in the space of synthetic biology, which is the science and engineering research that advances building with biological systems. And this really has tremendous opportunity to impact many types of products that can help society, everything from new types of medicines to agriculture to sustainable chemicals and materials, even new types of DNA storage. And it really comes from this idea of being able to put together biological systems or build with them from the word synthesis. And of course, those biological systems are frequently living or biotic of life. Increasingly, we as a society are relying on products made by biology to address many needs in planet and societal health. This includes things like new compostable materials made from mushrooms. It includes, for example, fibers that can be produced from carbon in the atmosphere and ultimately woven into clothing. It also includes new types of medicines like RNA vaccines, and even has important implications in the future of food, as shown here. Now, one of the challenges around synthetic biology and biotechnology more broadly is oftentimes when we produce the products of interest, we manufacture them in large manufacturing plants, such as the one shown here. And these manufacturing plants can cost up to billions of dollars to make. And one of the wrinkles is that oftentimes getting the product from these manufacturing plants to the places where it needs to be can require cold chain, certainly in the context of medicines. And so one of the challenges with the existing strategy for how we produce products by biology is this issue of both cost and distribution. And so my group is thinking about how can we create new forms of distributed biotechnology enabled by something called cell-free systems, which I'll give you a definition about in a few minutes. And this is really across multiple fields, from medicine to sustainability, allowing us to design biological systems faster and even education. So before I get started with a couple of vignettes around how we're using synthetic biology for people and planet health, I want to introduce, for example, what I mean when I say a cell-free system. So in my own group, what we do is we take living organisms or cells, such as shown here, we grow those cells up and we open them and collect their insides. And then what we do is we take their insides to, for example, produce products like a new vaccine or detect lead in drinking water, which I'll show you about a little bit later. You can kind of think of this like taking a car, lifting the hood up and pulling the engine out. It allows us to repurpose the insides of the cell to enable a whole new form of decentralized or distributed biomanufacturing. So just to kind of show this in a cartoon format, once again, what we do is we take cells, we rip off their cell walls and collect the insides. And this has all of the molecular machinery of the cell, but isn't living. And this allows us to then convert things like substrates of biology, energy, and information into products that we might use. And these systems are not genetically modified, they're programmable, they have low to no power requirements, they're shelf stable, and actually also can be distributed. You can think about like freeze-dried ice cream or freeze-dried fruit. And so what my group is doing is we're using these cell-free systems and applying them in a variety of ways to impact society. And so I'm going to show you a few vignettes, starting with protein medicines. So I think as many of you are probably aware, protein medicines have revolutionized our ability to prevent and treat human disease. We produce products like antibodies or vaccines, hormone replacement therapies, hormones or enzyme replacement therapies. And this has really had a transformative impact on the lives of many of patients. Unfortunately, the current manufacturing paradigm really limits our ability to distribute protein medicines. What we do is we tend to manufacture proteins in these large centralized facilities, something I introduced a few minutes ago, such as shown here, that can cost up to $1 billion to make. We then cultivate living organisms in large stainless steel tanks. These can be up to 10,000 liters in size or 50,000 liters in size. You can think of an Olympic-sized swimming pool, for example. And then we overexpress and purify the product, let's say an antibody therapeutic, an anti-cancer drug, and then we have to deliver these to their point of use, for example, a hospital. And key limitations around this strategy is the expense and time required to build such facilities and the feasibility of refrigerated supply chains to get medicines to where they might need to be. So my group, along with many others, are really interested at the moment in trying to imagine a new form of low-cost, decentralized manufacturing platforms that could broaden access to medicines by allowing us to make them when and where they're needed. As it stands, right, only up to 40% of the world's population lacks access to essential medicines just because we can't get the medicines to where they are needed. And so in terms of an example, I'd like to share some of the work we're doing around the idea of producing new types of antibacterial vaccines. So by 2050, it's now projected that more lives might be threatened by antibiotic-resistant bacteria than are threatened today by cancer. And so this really motivates the need to come up with new types of ways to address rising antibiotic-resistant bacteria. Conjugate vaccines are one approach to doing this, and they can prevent life-threatening bacterial infections. And the way they work is they teach our immune system to recognize pathogenic bacteria before they get inside of us. And that is based on two features of these. So on the outside surface of bacteria are these sugars. And these sugars act as a fingerprint to teach our immune system to recognize bacteria. And so what we do is we take these polysaccharides or sugar antigens, and we attach them to what's known as a carrier protein, or it's a protein that our body already recognizes as foreign. You can think of like a diphtheria toxin protein or a tetanus toxin protein. Many of us have had DTaP vaccines, for example. And so the idea then is that you're linking a sugar that's typically on the outside surface of bacteria to something our immune system already recognizes as foreign, and that can actually create an effective vaccine to prevent bacterial infection. And there are multiple examples commercially available. One that many people have heard of is Prevnar, for example, against pneumonia, that work. Now, one of the challenges with our current technology for making these conjugate vaccines is they involve multiple complicated steps, and they create heterogeneous products, reducing access. So for example, what we typically do is we grow the pathogenic bacteria up. They have these sugars on their outside surface. We extract those sugars, so we kind of strip them off of the outsides of the cells. We then put them in a form that they can be connected to this carrier protein, which is typically produced in another manufacturing step to ultimately lead to this conjugate vaccine, where we have the sugar conjugated to the carrier protein. Now, the limitations of this technology, it's typically challenging and costly to culture these pathogenic bacteria. Manufacture requires this centralized and cold chain dependent approach. And these sugars, it turns out, are typically non-specifically attached to the carrier proteins themselves. And so these have canonically limited the ability to really develop some of these vaccines. And so my group, along with our collaborator, Matt DeLisa, set out to rethink how conjugate vaccines are made using cell-free systems. So what we decided to do is rather than this kind of multiple step approach you saw on the previous slide, to create bacteria that were glycosylation competent, and they could actually produce the bacterial glycan that would typically be on the outside surface of a bacteria that we can use to stimulate our immune system to prevent against a bacterial infection, as well as the carrier protein itself in a simple one-pot cell-free platform for using these cell-free extracts to make conjugate vaccines. And just to show you an example, we developed a process that will allow us to produce these conjugate vaccines in a simple cell-free system. So in particular, I'm showing here an example where we produced cell-free synthesized conjugate vaccines for and against a particular pathogen known as Francisella tularensis. We purified those vaccine molecules, and we carried out a mouse study where we gave a single vaccine dose and then 2 boosters. And then we challenged those mice with a dose of this Francisella tularensis bacteria. And what the chart on the right is showing is days post the challenge with the bacteria, and the left axis is showing % survival. And what I want you to take away from this figure is that the negative control, so if we just give the mice a buffer or a carrier protein without the sugar from the bacteria, without the polysaccharide antigen, we do not get survival. But in the context of the cell-free synthesized vaccine, we actually get 100% survival of the mice, showing that we've created a cell-free platform for producing a vaccine that's very effective. Now, if we try to step back a little bit and think about how this might fit in the idea of inexpensive, rapid production without a cold chain, we actually developed these cell-free systems to be freeze-dried and to allow us to then have a freeze-dried powder so that we could just add water to produce a vaccine. And if you look at the numbers, what it means is typically some of these vaccine doses are on the order of about 5 micrograms per antigen dose. We produce these products at about 0.4 grams of antigen per liter, and it takes about 2 hours is all to make these medicines from these freeze-dried cell-free powders that are stable at 50 degrees centigrade for more than a month. We've also developed the system so that it can produce a vaccine for about 50 cents a dose in the raw material costs. What does this mean? Well, if we wrap it together, in a single liter, so imagine a single carton of milk, we can produce 160,000 doses of a vaccine potentially per liter per day. And if you had 1,000 liters, so a meter cubed, you could actually imagine producing about 160 million doses per day using our approach. So that's the first vignette I wanted to share with you, this idea that we could use cell-free systems to open access to how we might make medicines in the future. Turning to a different challenge, rapid population growth, a rise in global living standards, and climate change concerns have really intensified the need for sustainable, low-cost production of bioproducts. And myself and many others have been thinking about questions around how can we reduce carbon emissions and how can we capture above ground carbon for reuse into products that we might use every day. And so working with collaborators, we've been trying to engineer an organism known as Clostridia, which can grow on industrial waste gas or biomass syngas to make carbon negative chemicals. What does that mean? That means we would try to produce a product such as, in this case, acetone that's used in polyacrylic glass or other types of plastics or products or even jet fuels that we might use in society. But where that chemical that we produced is coming from carbon in the atmosphere. So again, a carbon negative chemical, we're taking carbon out of the atmosphere and going to use this organism to take that carbon out of the atmosphere to produce this product of interest. Now, one of the challenge with trying to engineer, let's say, for example, this Clostridia organism that we worked on with our collaborators is that it's typically quite slow. So in order to, let's say, produce acetone as a product, we worked with our collaborators to identify the enzymes or the molecular catalysts that could transform carbon dioxide or carbon oxides in the atmosphere ultimately into this product, let's say, acetone. And so we identified several enzymes for many different organisms that exist in nature. And then we went ahead and we put those enzymes into a cell and demonstrated that we could go from acetyl-CoA to acetyl-CoA, acetyl-acetyl-CoA to acetoacetate to acetone, and ultimately produce the product we wanted. But one of the challenges when you engineer living organisms is that when you introduce pathways from that may not exist naturally in those organisms, unwanted byproducts can be observed. And so in this case, what actually happened is in addition to making acetone, we started producing these byproducts. You could think of them as like shunts, siphoning off carbon away from the products of interest into things like 3-hydroxybutyrate or isopropanol. And if we looked at this method, a combinatorial knockout of such a large matrix of genes just wasn't very feasible with in vivo workflows. And so what we did is we used these cell-free systems to rapidly parse through and identify which ones of these particular shunts were actually active so that we could remove them. Then we worked with our collaborators to successfully remove the genes that were responsible for shunting carbon away from our product, in this case, acetone. And so we took this base strain, which produced just a few percent of this acetone product. You can see it's producing a lot of this isopropanol or 3-hydroxybutyrate. And we made individual mutations in this strain to ultimately get to a very significant fraction of the product now being this acetone product that we were looking for. And using cell-free systems, we gained about a year in time savings compared to how it would typically be done. And this process was scaled up into kind of the pilot scale process at about 80 liters, producing product at about 3 grams per liter per hour. So this work was published in Nature Biotechnology a couple of years ago. And we ultimately observed greenhouse gas emission reductions using this approach. So we're quite excited by it. What does this mean for kind of the bigger picture? Our collaborators at LanzaTech, what they're actually developing are these large Clostridium gas fermentation plants enabled by these organisms that can take carbon out of the air and now produce 50,000, for example, metric tons of ethanol per year, mitigating tens of thousands of tons of carbon dioxide that would otherwise be going into the atmosphere. Our group has also taken these cell-free systems into a different approach. So we can take these freeze-dried powders and instead of producing vaccines, we could repurpose them with cellular machinery to enable environmental and health diagnostics. So it turns out biological systems are always sensing the world around us. And we've been curious as to if we could create inexpensive freeze-dried technology that will allow us to, for example, detect if our water is safe to drink. As many of you know, with a rising population, one of the challenges we will be facing is the availability of clean water. And one of the challenges around water is that you oftentimes can't see, taste, or smell the types of contaminants that might exist in water. And so what we actually did is we programmed these cell-free systems in a freeze-dried system with a DNA that would allow us to run a genetic circuit and turn on expression of a colored product you can see here in the presence of lead. And so we're trying to develop a technology that for a very inexpensive amount could develop and detect lead in drinking water. I think as many of you know, lead in drinking water remains a major challenge, certainly in the United States. So we've actually repurposed these cell-free systems into other avenues as well, such as in innovating education and scientific literacy. So we've developed these freeze-dried cell-free systems to not only produce vaccines or detect lead in drinking water, but also to be able to educate the next generation of students and get them excited about science, being able to put science in the hands of high school students, for example, and get them interested in thinking about synthetic biology. So working with Jim Collins's group, we developed a user-friendly, inexpensive approach to be able to teach cutting-edge topics in synthetic biology. And here's an example of being able to basically create a light bright system of different colored proteins where students were able to produce different pictures. And you can see this one team even played a game of Connect 4 where the red team actually won. So what I hope to share with you today was just how cell-free systems, or this ability to take cells, rip off their cell walls, and collect the insides and reuse that, are providing some new frontiers for biotechnology. They can be used, of course, to rapidly express and evaluate protein catalysts, such as our example with LanzaTech, where we identified genes that were responsible for reducing the overall production of acetone in a commercially relevant process. They can also be used for distributed biomanufacturing of next-generation vaccines, where, as I mentioned, we could imagine producing tens of thousands of vaccines in a single liter for about 50 cents a dose. They can be used in portable diagnostics, as I showed with this lead detection system, and even in trying to widen the access to new education models for synthetic biology. So before I close, I just want to mention that a lot of the work we're doing has been supported by the federal government, has been carried out by a ton of wonderful students, pictures of which are shown here. So thanks again, Piero, for the fantastic opportunity to be here, and I'm happy to try to address and answer any questions from the audience today.
Yeah, I have a question that you already answered in your talk. But if you go back a few years, I think there was, let's not call it consensus, but there was the idea that cell-free systems are not suitable for large-scale applications. And I think because of the yield, essentially. Don't know if there were other reasons. So first of all, is the perception changing in general? I mean, of course, in your research group, but in general, in the industry. And second, what if it is changing? What has really caused the change in perception?
Good question. So the question is really about the industrial applicability of cell-free systems, and do we see a new wave of interest in them, certainly from a large-scale production perspective? So I can answer this in two ways. So in the first way, over the last 5 years, we've continued to see reductions in cost of cell-free systems as well as increased capabilities, both in the context of producing, for example, glycosylated vaccines, like I showed you today, but also in these diagnostic systems that are becoming accessible at really low costs, $1 a test kind of examples. And so what we're seeing is industry's gotten very excited about that, but it's still a new manufacturing approach. And so what I would say is there's an increasing wave of interest, certainly from pharma partners. So I have one of my startups, for example, has a partnership with a pharma partner who's gotten excited about thinking of different manufacturing approaches. And so I think there's a lot of interest gaining for using cell-free systems at scale at the 100-liter, 1,000-liter, or even larger scale, certainly for both protein medicines and small molecules. Separately, there's even bigger wave of interest in using cell-free systems to kind of prototype engineering designs or biological engineering designs, because we can generate data much faster than is typically done inside of cells. And as a result of that, cell-free systems are being particularly embraced by the industrial community and commercial community for generating data to inform machine learning-based models for accelerating biological design. And so we've seen that particularly around protein engineering, but as well as other types of strategies.
So that's also interesting. You mentioned data, you mentioned machine learning. So I know nothing about biology, so forgive me, the questions are silly. But does that mean that cell-free systems are also somehow more suitable than cell-based systems for this computational biology that we live in, basically?
So I think there are pluses and minuses to both living systems and cell-free systems for generating data at high throughput. But what I would tell you is for example, you can do selections inside of cells, like live dead selections that might otherwise not be possible in cell-free systems because you don't have the live dead question. But what I would tell you is from a rapid perspective, you can generate DNA, and your process in cell-free systems can go much faster from DNA to protein than is in a cell, because you can buy DNA, have that delivered from a DNA synthesis provider. Even in linear expression templates, you don't need to do any cloning, putting that DNA back into a cell, and you can express your protein. So for example, we worked with David Baker's laboratory on developing some antiviral protein therapeutics against COVID. And so his group would develop and design these on a computer. We would then buy the DNA, and within hours, we would have the proteins to be testing them as to whether or not they were going to be a more efficacious binder against, let's say, the SARS-CoV-2 trimeric spike protein. And so we didn't have to put them in cells or wait for transformations or many of these other steps. The other really interesting thing for speed in terms of generating data is that cell-free systems, because they're not living, you don't have to grow them and keep them alive. And so you reduce a lot of the manipulations to automated liquid handling. And so my lab actually does a lot of work with acoustic liquid handling, where we use sound to move liquids very rapidly in order to assess, higher dimensionality of space. And so we're very excited about this idea of using cell-free systems for rapidly generating data for machine learning-based models. We recently had a paper about a month or so ago in Nature Communications. That's one example that I could point people to, with Landwehr as the first author, but there are other examples as well.
Fascinating. We could talk a long time about this. Anyway, you have a question in the chat. How stable are the conjugate molecules? If the sugar label is lost, are the remainders toxic?
Yeah, so good question. So how stable are the conjugate molecules themselves? So you can imagine actually that the proteins could be oxidized, or there are other things that could happen to the carrier protein itself that could make that less effective. You can also have the sugar being cleaved off of the protein inside of the cell in a therapeutic regimen. And so that is certainly a concern. And if it's just stable or sitting for a long time, you can also have instability issues. And so that's one of the advantages of cell-free systems. We can stockpile all of the reagents and within 2 hours make the conjugate vaccine, and then imagine being able to dose that very rapidly. Your question about the carrier protein being stable, oftentimes these carrier proteins are already something that we've used to elicit an immune response to protect against something else, let's say tetanus or diphtheria, other types of pathogens. And so on their own, they're fairly safe molecules. They're things that have been in the clinic and in humans for many years. And so that has not been a major concern.
Okay, excellent. You're lucky we're not in a physical room, because I would keep you captive for much longer. And
well, wonderful. Thank you for the questions. And it's really been a pleasure to be here.
yes, thank you. I know you have to go.